Hello, I'm Ruslan Medzhitov. Today I will talk about the role of gut-brain axis in food allergy. All diseases are abnormal versions of some normal physiological process of function, and to really understand a disease, we need to know what the normal physiological counterpart is. And in particular, many diseases are due to excessive or insufficient expression of various body defenses. In the case of allergies, I think the normal counterpart of allergic disease is what I refer to as allergic defense. Allergy is typically thought of as a mistargeted pathological response to innocuous environmental substances, such as food proteins. However, all allergic reactions exhibit the features of defense systems that protects us from noxious substances. All symptoms of allergy, such as running nose, sneezing, itching, vomiting, diarrhea, have to do with expelling something from the body. So therefore, this defense from noxious substances, I think, is the normal counterpart of allergic disease. So in the case of food allergy, the normal counterpart would be, again, something that protects us from potentially harmful substances in the food. So food allergy is a reaction of the immune system to food substances that our body, for one reason or another, deems to be noxious, harmful to us. And normally food allergy is a part of what I call a food quality control system that includes olfaction, taste, and various gastrointestinal chemosensing mechanisms. And pathological food allergy then would be an overreaction of this quality control system. The quality control for food is important because food is not just nutrients. All food sources, natural food sources anyway, contain a variety of chemicals. Some of them are innocuous, some of them are useful for our bodies, and some of them are harmful. And how our body deals with this huge diversity of thousands of different chemicals is largely unknown. And here on this slide is just a snapshot of some of the chemicals identified in some of these common foods. And for the most part, we don't know how any of these molecules are perceived by our body, whether they actively sense, and what are the consequences are of exposure to many of these chemicals. And plants may make many chemicals for defense purposes to protect from being eaten by herbivores. And so a lot of plant secondary metabolites are actually designed to be toxic to animals. And therefore, it's very important for animals that consume plants to be able to detect these noxious chemicals and to react accordingly. And plant-derived chemicals is only one example of this rich diversity of molecules that are present in normal foods. And we actually know very little about the full universe of chemistry that we are exposed to, both natural chemistry as well as chemistry that is a product of industrial production, such as the one that's present in modern foods during food processing, in detergents, and other chemicals present in normal household items. So the way that food quality control works, starting with more familiar examples, before we even consume something, it has to pass a smell test. So if it smells fishy, if there is something we don't like about the smell, we don't ingest it, obviously. But if it smells okay, we proceed with ingestion. And at that point, the gustatory sensory system detects the quality of food based on its taste by detecting various molecules present within the food. And again, if it tastes in a way that is suspicious to us, we don't proceed with ingestion. But if it tastes fine, we swallow, and then the food gets into the GI tract, where there is another chemical sensing system that evaluates the quality of food. Most of what we know about this chemosensing system is based on recognition of macronutrients, proteins, carbohydrates, and fats. And there are a variety of specialized sensory epithelial cells that detect these macronutrients and report their value to the brain and to endocrine cells through production of various gut hormones. This communication is very important for adjusting our metabolism, satiety, hunger, cravings, and so forth. So this is a very complex system that communicates between the gut and the brain. And in this case, it's directed at the detection of the nutrients. But in addition, there are specialized sensory cells, including enterochromaffin cells and Tuft cells, that are specialized on detecting potentially harmful substances in the food. So this would include various toxins, noxious xenobiotics, and so forth, and as well as some of the infections, including parasite infections. And these two types of cells then, when they detect noxious chemicals, also communicate to the brain and to the rest of the GI tract to elicit appropriate reactions. So if we look closer at how the food is being sensed, there are these different types of sensory epithelial cells, including enteroendocrine cells, Tuft cells, enterochromaffin cells, as well as just below the epithelial layer, there are immune cells and various types of neurons that monitor also what's going on in the gut lumen. Enteroendocrine cells, again, they detect macronutrients, amino acids, sugars, and so forth, produce these gut hormones that communicate with the nervous system and endocrine system. Enterochromaffin cells and Tuft cells detect noxious substances and produce, again, various signals, including serotonin, as well as some of the cytokines like IL-25 and TSLP that then can communicate with the immune system in the gut. And what happens at this stage is what's largely unknown in terms of the mechanisms resulting in activation of the immune system. But detection of noxious substances on the one hand and detection of dietary proteins on the other hand creates these two requisite signals that are necessary to initiate the adaptive immune response. In this case, Th2 response that can result in allergic sensitization. This Th2-driven or so-called type 2 immune response, we think, participates in food quality control. It evaluates food composition and it promotes defensive reactions that meant to minimize exposure to noxious substances. This would be mediated through IgE antibodies recognizing dietary proteins, inducing mast cell degranulation, release of histamine, induction of peristalsis that would promote expulsion of the noxious substances, as well as through activation of C2 cells, production of IL-13 that will promote mucus production, increasing barrier function, and again protecting us from noxious substances. And as you can imagine, exacerbation of these reactions would result in a typical pathology of food allergy. So we were interested in the question of how the brain reacts to these allergic responses in the gut. It's been known since early 90s, mice that are sensitized to a particular type of food, for example, to corn or peanuts, once they sensitize, they avoid consuming that food. We asked whether this behavioral response to food allergy represents a form of associative learning. Associative learning, as you all know, exemplified by this classical conditioning, Pavlovian conditioning, where an unconditional response or pre-programmed stereotypic response to a stimulus, like in this case food, and there would be no response of the same type to a conditional stimulus, like the sound or something else that is not related to food. However, when these two stimuli are paired, then there is an association between them, and that's what results in associative learning or conditioned response to conditional stimulus alone. So that's a typical sort of Pavlovian conditioning. And there is an example of Pavlovian conditioning that occurs in the phenomenon of conditional flavor aversion, or sometimes called conditional taste aversion. And what happens here is that if you consume food that contains a noxious substance like a toxin, the toxin itself will result in food poisoning, which would be an unconditioned response to a toxin. So to avoid consuming the same food containing this toxin in the future, what happens is that because we cannot see or smell the toxin typically, but we can detect the flavor of the food that contained the toxin, then what would happen is that the flavor of the food would be associated with the malaise induced by toxin, and that association will result in long-term memory towards the flavor associated with the food in order to avoid potential consumption of toxin in the future. So the flavor now will operate as a conditional stimulus, resulting in conditional aversion of the flavor or the taste. And in this way, the system minimizes the risk of exposure to toxin in the future. And I think what happens in food allergy is very much parallel to that, except that instead of the flavor of the food, what is being detected is food antigens. Because the immune system, in order to react and remember what it reacted to, it needs to see protein antigens. So food antigens are used as a conditioned stimulus, and when they're present by themselves, it elicits no response or immune tolerance. When it's present together with a noxious substance that can induce adverse effects, then there is associative learning between the dietary protein antigen and the noxious substance that results in allergic sensitization, and the next time we expose to the same food with the same food antigens, it will result in conditional diversion. Of course, it will result in a normal allergic inflammatory response in the gut, but we hypothesize it also will result in behavioral response of aversion. We found evidence for this framework in a few experiments that I'll share with you. The experimental design here is very simple. We use mice that are sensitized to a particular antigen, like ovalbumin, and then we see whether they exhibit aversive behavior. But first I'll just show you the control scenario where these mice are not sensitized. So they have a choice of consuming either water, pure water, or water containing the ovalbumin, which they're not sensitized to. So it's not an allergen for them yet. And as you can see, mice actually prefer to drink water with ovalbumin. It probably tastes better for them. So they consume a lot more ovalbumin-containing water in two different genetic backgrounds of mice. And this is shown in two different ways, either preference for one bottle versus the other, or by the number of times they lick the bottle, as shown on the right side. So they control ovalbumin-containing solution over pure water. However, once they get sensitized, once they become allergic to ovalbumin, then what happens is that they stop preferring the solution containing ovalbumin, and they start preferring water over ovalbumin containing solution once they become allergic. So that's what it means that they become aversive to food or water sources containing the allergen. And what is particularly interesting is that this aversion is entirely dependent on Ig antibodies. So this behavioral response depends on the presence of antibodies towards the antigen the mice avoid consuming. So this is shown in a couple of ways here. This is comparison of wild-type mice. When they're not sensitized, they prefer ovalbumin, and when they're sensitized, they prefer water. And this preference goes away if they don't have Ig in Ig knockout mice. And similarly, if they don't have Fc epsilon receptor for Ig, they also lose their aversion towards ovalbumin. And this is shown also here using a different type of readout. And interestingly, this type of aversion is very robust and stable. It can last for months. So here we show the comparison again between control mice that prefer ovalbumin versus allergic mice that lose this preference and start avoiding ovalbumin. This is 10 weeks after sensitization, and this is over 50 weeks after sensitization. There is still the same phenomenon. There's a long-lasting memory of aversion. And this aversion reaction is actually not dependent on inflammatory response in the gut. As shown here, this is the control gut, normal mucus of the intestine. This is allergic mice that are wild type. You can see a lot of goblet cell hyperplasia, mucus production. And this is Ig knockout mice that also have hyperplasia and mucus production. However, they are no longer avoiding consuming the ovalbumin in the absence of Ig. What we found is that this sensing of allergen in the gut leads to activation of sensory neurons that result in activation of nucleus of solitary tract in the brainstem that then connects to parabranchial nucleus that monitors various sensory information from visceral organs, and then in turn communicates with lateral hypothalamus and amygdala, where the memory of this experience is stored. So this gustatory, olfactory, and chemosensory information from the gut goes to the nucleus of solitary tract that collects all this information, transmits it to parabranchial nucleus, where this conditional taste aversion and adverse reactions to food recorded, then transmitted to lateral hypothalamus, and to amygdala to induce aversion and anxiety, which is also present in human patients that have food allergies. So this is an example of gut-brain connection where the sensory neurons that monitor the gut and connect to the brainstem actually get activated not directly by a stimuli from the gut lumen, but are activated through IgE antibodies that recognize allergens. And IgE antibodies then get deposited on mast cells. Once they recognize the allergen, leads to mast cell degranulation, release of histamine and neuropeptides that then activate the sensory neurons, leading to the activation of those brain circuits that I just showed you. So this is an interesting variation on the theme of how neurons detect various stimuli. They can either detect them directly, as is the case with olfactory neurons, or they can detect them via specialized epithelial cells, as shown here for visual system and auditory system. And here we now have an example where it's stimuli detected indirectly through antibodies that sit on mast cells and induce the degranulation and release of neurotransmitters that activate the neurons. In conclusion, conditional allergen aversion is a version of conditional taste aversion, except that instead of flavor or taste, what is being detected is food antigens, and that results in a very similar type of aversive behaviors. Allergic sensitization results in allergen avoidance, which is a behavioral defense against consuming noxious substances, and it's IgE mediated and involves these brain circuits that generally involved in defensive behaviors. Finally, this work was done by a very talented postdoctoral fellow, Esther Florsheim. She was helped by a very talented research associate, Jamie Cullen, and Fernando Carvalho assisted with some of the experiments. And two collaborators, Marcel Dietrich and Daniel Nisida, helped with various aspects of the project. And this work was supported by Food Allergy Science Initiative and HHMI. Thank you very much for your attention.